Welcome to Ask the Expert. Today we have Dr. Cristina Nostro coming to us from the McEwen Stem Cell Institute in Toronto. Her talk is titled Building Human Islets from Pluripotent Stem Cells. She is a senior scientist at the McEwen Stem Cell Institute at University Health Network, associate professor at the University of Toronto, and holds the Harry Rosen Chair in Diabetes Regenerative Medicine Research. She received her PhD in biomolecular scientist from the University of Manchester in 2004. Her work is focused on the generation of pancreatic islets from human pluripotent stem cells with the goal of developing novel treatment for type 1 diabetes. And we're very excited to talk to her a bit today. She's going to walk us through her recent work, and then hopefully we can touch on her recent Science Direct review, which was designing next generation islet-like cells from human pluripotent stem cells, advances in overcoming allo rejection and recurrent type 1 diabetes. And that was released recently as well in 2022. But that, and that really kind of, aims at one of the biggest questions is how can we implant these cells into humans successfully. So we're really excited to hear from you. Thank you so much for joining us on World Diabetes Day, November 14th.
Thank you so much, Monica, for that really lovely introduction and, the invitation to present at the Sugar Science. And it's especially on this special day of World Diabetes Day. So I would be remiss if I didn't remind everybody here that diabetes is a global emergency, is affecting over 500 million people worldwide, and this number is destined to increase. And while some patients with type 2 diabetes, for some of them, diabetes is preventable. Type 1 diabetes is not preventable. And people living with type 1 diabetes suffer from an autoimmune disease that attacks the beta cells and destroy them. And so the first line of treatment is exogenous insulin administration through pumps or needles. So recently, FDA approved immunotherapy to delay stage 3 type 1 diabetes, and this is, fantastic for some individual at high risk. But realistically, we don't have a way of preventing or cure the disease. The closest we get to a cure is pancreas and islet transplantation. But these type of treatments are only offered to selected patient population, usually adults, which are undergoing other transplant, like typically kidney transplant. So they're already on immunosuppressants. But because islet and pancreas transplantation work, we have, the premises for the work that we're doing. So generating surrogate islets from stem cells. They work, but they have certain limitations. First and foremost is the limited supply of donor organs, the declining in graft functionality, and the most important aspect, which is the requirement for chronic immunosuppression. Monica mentioned a review that we recently published. I'm not going to touch on this topic today, but it's certainly an area of, intense investigation at the moment. I think, because these are limitations that are affecting pancreatic and islet transplantation, but these are we live in a remarkable time, because I think with the use of pluripotent stem cells we could address all of these limitations. This population of cells have the ability to give rise to all the cells in the body, and so by harnessing this capacity, we can actually manufacture surrogate islets and develop potentially a consistent product, both in quantity and quality. And quantity is like one of the major limitations with islet transplantation. The other limitation is the fact that when we transplant islets, the majority of the islets that are transplanted, about 50 to 60%, die in the perinatal, transplantation time because of the lack of a good connection to the host. So by improving the early connection to the host, we can actually improve survival of the cells and have a better possibility to have long-term functionality. And so there is a lot of studies that are looking into that. We did some work on the use of microvessels to promote vascularization in 2021, and really showed how remarkable is, what you do earlier on in the transplant can affect the functionality of the cells. And last but not least, if we want to generate a product that could be used for the majority of people with type 1 diabetes, we'll have to eliminate immunosuppression. And so using pluripotent stem cells is offering us the opportunity to use both bioengineering and genetic engineering to protect the cells and eventually generate a product that could be transplanted without immune suppression. And so how do we generate this consistent product? This will be pretty much the topic of today's talk. There are several approaches. Have you heard in previous talk of different ways of making islets? What I'm going to focus today is directed differentiation. And by directed differentiation, I'm essentially focusing on the way of mimicking what happens during embryonic and fetal development. So what we're trying to do in vitro from pluripotent stem cells is trying to reproduce those developmental steps and generate a specific tissue or cells. And so we've learned from several model organisms, going from zebrafish embryo, xenopus, the mouse, on how these organisms develop a pancreas. And we know that the steps that are involved, and this is what you see here is a schematic, being the first step is the generation of the definitive endoderm, the germ layer that will give rise to the entire gastrointestinal tube and all the organ associated with this tube. And so we have to be able to generate definitive endoderm and pattern this tube into the pancreatic progenitor population. And so you can see it here kind of depicted in orange. This is the area where the pancreas will form. These cells will express PDX1. And when they commit to the pancreatic lineage, they will co-express another transcription factors known as NKX621. So it's really the identification of these two markers in vitro that gives you an idea that you are on the right path to make pancreas. So these pancreatic progenitors will then give rise to both the endocrine and the exocrine lineage of the pancreas. And it's really through, the work of many, many people in this field that in the past 20 years, we've actually learned how to really control the differentiation and take pluripotent stem cells to these developmental stages, going through the definitive endoderm that I mentioned before. The germ layer gives rise to the gastrointestinal tube that expresses PDX1, then the pancreatic progenitors expressing PDX1 and NKX621. And in 2014, two groups really pioneered the development of the hormone-positive cells from these pancreatic progenitor cells, a group of Doug Melton and Tim Kiefer, showing that you can generate hormone-expressing cells in vitro. And typically, because we're interested in, particularly insulin-producing cells, we monitor for the expression of C-peptide as a surrogate marker for insulin, and NKX621. And what you see here in this flow cytometric plot is the co-expression of C-peptide and NKX621, depicting what we call beta-like cells. And, because of this tremendous work, and 20 years, which I think it's remarkable, we've now taken these cells to clinical trial. The initial trials were transplanting the pancreatic endoderm, those cells that I told you are progenitors, so they express both PDX1 and NKX621, and recently the islet-like cells that are transplanted intrahepatically, yes. And what we are learning from this initial trial is that the pancreatic progenitors transplanted subcutaneously generate insulin-positive cells in patients with type 1 diabetes, but the beta cell mass is not sufficient to normalize glycemia. So we would need to transplant many more devices and large number of cells to and probably also figure out a way to support the vasculature at the time of transplantation much better than we are currently doing to really have something that could normalize glycemia. while the use of a more mature population, and I'm cautious here on the more mature, but definitely more hormone expressing cells, they're, in a way, surrogate islets transplanted through the portal vein into the liver. We are actually seeing remarkable data suggesting that these cells can actually lead to the elimination of insulin, exogenous insulin in this patient, and normalizing glycemia. However, if these are to be transplanted like cadaveric disease donor islets, they will suffer for the same type of issues. So the fact that at the time of transplantation, you don't have a close connection to the host vasculature, so a lot of these cells will actually die. So we need to promote engraftment at the time of transplantation. We need to eliminate rejection. This particular trial is running with immunosuppressant. And last, which I think it's an important topic, especially if we're gearing towards genetically modify the cells to immunoprotect them, we need to make sure that the cells are safe and possibly functional, or for the future. And so this is what I'm going to be talking about today: safety, focusing especially on safety and functionality. And I'm going to start with functionality. Many of those who are working in this field realize that what we're making in vitro, although we call them islet-like cells, they are actually not metabolically mature like primary islet. And while they sometimes release a similar level of insulin in response to glucose challenge, if one take a closer look at the metabolism of these cells and calcium oscillation in the presence of high glucose, they're quite different than adult islets. You see here the calcium oscillation of primary islet in response to high glucose are kind of missing in the stem cell islet preparation. So there is definitely still more work that needs to be done to further mature these cells in vitro for future use. I should also add here that maybe, generating a fully mature islet is not necessarily a requirement for functional in vivo treatment, because maybe an immature islet might have less requirement for oxygen and nutrients than a mature islet would. And so maybe it is an advantage to transplant something that is immature, but definitely this is something that needs to be elucidated. Currently, even in clinical trial, the islets that we're transplanting require several months before they produce enough insulin to normalize glycemia. And work by Jeff Millman has demonstrated beautifully that these cells would take approximately 6 months in a murine model to acquire a transcriptional profile that resembled mature islets. So there is definitely, a black box here that we can investigate.
Sorry, Cristina, that work by Jeff Millman is beautiful. I have a question here in the chat from Boston. Would these islets show the same phenotype in vivo?
Yeah, this is a good question. I actually, I don't know that this experiment has been done. I don't know that people have taken the islets transplanted, if this is a question, like the transplanted islets, and then isolated after the transplantation, whether they would have a profile that more resemble the deceased donor islets. We haven't done it ourselves. Not on the calcium oscillation. Yeah,
it's pretty tough to get those recordings of measurements in vivo.
Yeah, you can do, you would have to isolate them, I think.
Mm-hmm.
Sorry, I don't see the chat, so please stop me anytime if there is important questions throughout.
Yeah, sounds great. I'm here as the moderator.
Yeah. The other question that we have about the other difference, if you want, about the stem cell derived islet compared to diseased donor islet is that they also contain off-target populations. So one of these particular that is, kind of a hot topic at the moment is the enterochromaffin cells. So you see here, this is single cell analysis performed by the group of Doug Melton in 2019, who first identified this population, showing that you have your beta cells, the purple cells, alpha cells, and also like a large cluster of cells that we called enterochromaffin cells. These are not typically found in the islet. They're cells that are found mostly in the intestine, and if anything, they're taking up space, right? So if you want to transplant something that could have a therapeutic effect, you want to try and maximize the number of cells that are pancreatic through pancreatic. So definitely something that we are looking into and trying to remove them from our culture conditions.
I don't see the delta cells in there.
Yeah, actually there are. I don't see them either, but we have done single cell analysis of our end stage culture, and I know from the other studies that I'm listing here on the right that many people have actually identified somatostatin positive cells from this population. So you're right. I don't know why they're not there, but we know for sure we're making somatostatin. Yeah.
Oh, thanks. We've got the feedback. HHEX positive cells are the deltas. Thank you.
Oh, sorry. Yes.
That's great. Thank you.
Sorry. So it's amazing how sometimes you look for something written in a specific way. So to try and address and trying to optimize this, in a way, our end stage population, Adriana Migliorini, a senior postdoc in the lab, decided to perform an analysis of the human developing pancreas. Because, if you remember what I told you in the beginning, the way that we will learn how to control the differentiation of human pluripotent stem cells is by looking at studies in various model organisms. And while there is a lot of similarities in pancreas development between mouse and humans, they're definitely not the same thing. So Adriana set up to obtain tissues from human pancreas during the second trimester, spanning between 14-week to 18-week post-conception, with the idea that during this particular time of development, there is an extensive islet formation. The islets are being remodeled, and you go from small islets to larger clusters. And she dissected the tissue, isolated the nuclei, and performed single nuclear RNA-seq using the 10x genomics platform. And this was done in collaboration. Yes, this was done in collaboration with the analysis of this data was done in collaboration with the group of Fayyaz Nota, the Princess Margaret Cancer Research Center here in Toronto, and particularly with a remarkable PhD student, Sabrina Gay. And Adriana and Sabrina worked together to analyze this population. They were able to annotate over 70,000 cells, and you can see the classical population that you would expect: the acinar cells, the ductal cells. I don't know if you see the mouse, the endocrine population. And particularly, Adriana was interested in identifying the niche of the pancreas. You see the mesenchyme, immune cells, endothelial, neuronal cells. And we know a lot about, the crosstalk between the mesenchyme and the requirement for the mesenchyme for pancreatic development. But what was, in a way, less studies is the role of the immune cells during pancreatic development. And this was, a topic very dear to Adriana, because she's also interesting in how, and elucidating in a way how the immune cells not only play a role in pancreatic development, but also may also promote the development of tolerance during these early stages of development. And so together with Sabrina, they focused on these immune cells and resubsetted these 5,000 immune cells that they identified in the in the pancreas. Sorry, I'm a little bit slow here on the arrow. Okay. And so these are the 5,000 immune cells that they isolated, and they were able to annotate all of them. And like you can see, you know, they identified T cells, T cells, NK, B lymphocyte, erythroid, macrophage, and monocytes. And why we were not surprised to see you know, different type of T cells. We see, you know, CD8, CD4 T cells. We see different type of B lymphocytes. This was kind of expected, but what was surprising to us was the identification of different subsets of macrophages. And Adriana took a closer look at the macrophages, and she annotated them as the perivascular macrophages for the expression of CD36, LIVE1, and MRC1. You see them here on the top, while this other group of macrophages that is here, that cluster 12, was actually characterized by the expression of TMD4, MMP9, and IL-18. And because of the expression of this marker, Adriana defined them as phagocytic macrophages. At the time of this work, she also compared her data sets with the data sets generated in Germany by the group of Christian Conrad, the first author is Tosti, to compare her macrophage population to the neonatal and postnatal macrophages. And we were not able to identify a similar subset, which was to us, and I, you know, the first, you know, thought to speculate that maybe these tissue resident macrophages actually are developmentally controlled, and they are unique to the developmental pancreas, while, you know, they may change phenotype during postnatal time. And intrigued by this marker, she wanted to verify that these proteins were indeed expressed, and she performed immunofluorescence analysis. What you see here is cytokeratin 19 marking the ductal epithelium, or the pancreatic epithelium, I should say at this point, in cyan. CD36, which marks typically endothelium, but as you can see here by the expression of IBA1, which is a macrophage marker, some of the macrophages are actually co-expressing CD36, as shown by the single nuclear RNA-seq. And importantly, they are close to the vasculature and the pancreatic endothelium. Here you see other macrophages expressing, in this case, we're looking at CD68 as a marker of macrophages, closer to an islet that express C-peptide. And some of these macrophages co-express LIVE1, some do not, you see here, possibly an indication that some of them are the phagocytic, and some are the perivascular macrophages that Adriana identified in the RNA-seq analysis of the pancreas. And obviously, because of this presence of macrophages and the concept that we know tissue-resident macrophages play a tremendous role in tissue homeostasis, tissue remodeling, Adriana speculated that maybe they could play a role in endocrine differentiation. And so we were very fortunate because we work closely, physically close to Dr. Gordon Keller, who's one of the leaders in hematopoietic cell differentiation from human pluripotent stem cells. And Gordon Keller, in collaboration with Michael Atkins, who was a PhD student in his lab, developed a protocol to efficiently generate what we call the primitive macrophages. So hematopoiesis is well known to come in different waves. The first wave, the primitive hematopoiesis, gives rise to cells like primitive macrophages that can actually seed the developing organ and give rise to tissue-resident macrophages. So it is, at least in mouse, it has been shown that many of the tissue-resident macrophages have a primitive yolk sac origin. And so Gordon Keller has been able to reproduce this differentiation in vitro and generates what we call primitive macrophages. And
just to back up 2 slides, I think a question: are there any blood vessels close to the islet in this context?
So what you see here, the live one here, that is not that is not CD68 positive would actually identify the endothelium. So probably yes.
Yeah, good qualifying question. Thank you.
Okay. And so we were, so Adriana wanted to develop a co-culture system to study whether endocrine development could be supported by the presence of these macrophages. And remarkably, these primitive macrophages actually express, the generation of these macrophages is very efficient. They express typical markers of macrophages, but most importantly, they express CD36, as you see here, and MRC1, two of the markers that we identified in the single-cell RNA-seq analysis. And so Adriana developed a co-culture system to study endocrine development in the presence of macrophages. And, I'm summarizing in 2 slides, a lot of work, but she essentially set up this culture where she mixed the human ES-derived primitive macrophages with developing PDX1 positive cells. And at first, the very first question that she wanted to address was whether the presence of macrophages impacted the development of the pancreatic progenitors. And what you see here in the bottom is the flow cytometry analysis for PDX1 and NKX611. You can see that she generates a similar frequency of PDX1 positive NKX611 pancreatic progenitors and similar numbers. So the presence of macrophages does not change the differentiation of the pancreatic progenitors. But interestingly enough, as we move this culture further, and typically we give rise to the formation of 3D aggregates to push the endocrine differentiation. The macrophages start to surround the endocrine clusters. You can see here the picture on the right in pink is CD45, and the majority of the CD45 positive, not the majority, I should say, all the CD45 positive in the cells in this culture are macrophages. They're, wrapping up the endocrine core. And the other remarkable facts of this culture is that when we co-culture the macrophages with the endocrine cells, the cultures are very clean. And when by clean, I mean you don't see any floating dead cells in this culture, most likely because the macrophages are phagocytosing all the apoptotic cells in this culture. While most of the macrophages are outside, Adriana has shown that some of the macrophages also go inside of the core. So it's a much healthier environment. And probably as a result of that, we see like a remarkable production of endocrine cells. So in the presence of macrophages, Adriana is able to generate higher frequency of pancreatic endocrine cells, the NKX611 chromogranin positive cells, and higher yield. So it's not just frequency, it's also higher number. And so this, the presence of macrophages is, in our minds, promoting endocrinogenesis. So this is, I just want to stop here for a second, just as a take-home message. What we've shown so far is that we've identified a subset of macrophages in the fetal pancreas that seems to be distinct in its phenotype to postnatal macrophages. And based on our in vitro culture, it appears that these macrophages have a role in promoting endocrinogenesis. And so, of course, we're speculating that this might be happening also in vivo during development.
Fantastic. Just a quick questions, 2 questions from the chat. Any idea on the macrophage subtype M1 or M2? And is there any difference in M1, M2 macrophage differentiation during culture?
So these macrophages, I don't have the data to show you here in this presentation, but they've been analyzed in details by the Keller group, and they seem to be non-inflammatory. We've done some comparison to cord blood-derived macrophages. They express lower level compared to cord blood-derived macrophages of inflammatory cytokines. So the idea is that, we speculate, that the role of these macrophages is more to do with homeostasis, possibly promoting angiogenesis at the site of in normal development, and phagocytosing old apoptotic cells. Yes.
Got it. And then also, is the macrophage promoting differentiation from PP or proliferation of differentiated endocrine cells? Can you tell that from what you see?
Yes, so we don't see difference in proliferation actually, but we see an increased frequency of endocrine cells. So it seems like it's promoting actually the endocrine differentiation.
Mhm.
We haven't identified if there is a specific growth factors that is supporting that. Yeah, if that's where this question is going.
Yeah,
Yes.
I think that's where it is going.
What I'm not showing you is that the macrophages do not impact the endocrine lineage specification. So the frequency of beta cells, the frequency of polyhormonal cells, the frequency of enterochromaffin cells doesn't change. So we're not, unfortunately, the macrophages, unfortunately, are not skewing up one lineage versus the other, and for sure they're not eliminating this off-target population that I was telling you about before, this enterochromaffin cells.
Right.
Well, we have, yeah, sorry.
Then there's a share in the chat that's an interesting paper about M2, M1 macrophage on pancreatic endocrine proliferation that's shared in a suite to everyone if people want to check that out. And then from City of Hope, a question, are these macrophages mimicking the ones from yolk sac or bone marrow?
Yeah, so they're mimicking the yolk sac. That's why we call them primitive macrophages, and they're definitely not the definitive macrophage derived or monocyte derived macrophages.
Mhm.
Yes. Sorry, I'm just opening the chat as well so I can.
I feel very, very excited about this. There's a lot of questions.
That's great. It's good. Hopefully I can address all the points, but I'm happy to chat offline if I'm not, answering properly. And I'm sure Adriana would be super excited to talk to all of you. She's the expert, and I should have really done a much better job with this, and she should have been here. So it's my fault. I apologize.
No worries. Perhaps we'll have her in the future.
Yeah, exactly. She can come and give another talk for you guys. Yes. So what I was telling you about that is that, despite improving and promoting endocrinogenesis, we're not seeing changes in our efficiency of making only pancreatic cells. And which would have been nice, right? But what we have learned from the single cell analysis from Adriana is that indeed these enterochromaffin cells that we're making in the dish are an artifact of our cultural condition, because these are cells that are not normally found in human pancreas development. If you focus here on the endocrine population, and we look for the marker of enterochromaffin cells, which is SLC18A1, which codes for the VMAT1 protein that is expressed by enterochromaffin cells. We don't see it in the fetal pancreas. So we know that what we are doing, what's happening in vitro is definitely an artifact. And so we should be able to learn how to control this. That's my bottom line. Sorry. And so the question is really, are these enterochromaffin cells a problem for beta cell replacement therapies? We don't know what's being transplanted in clinical trials, so I can't, say whether these patients are actually, being transplanted with population of cells that contain this EC or not. But what I can tell you for sure is that in all the studies by our group and all the studies done by other groups, we always see them. So it's clearly there is something in our culture condition that is pushing this lineage to arise. What are these cells? I told you they produce serotonin. They're normally found in the intestine. They are important for promoting intestinal motility. high concentration of serotonin has been associated with irritable bowel syndrome and carcinoid syndrome. And we know that these cells have also been identified in pancreatic metaplasia, which are the early stages of pancreatic cancer. What I told you in the beginning is that even if they're inert, which might not be the case because early studies from the Melton group suggest that they might be inhibiting glucose secretion, insulin secretion in responses to glucose challenge. So we are trying to figure out a way or means to control this. And so one of the first questions is like, why are we making them? And our first interpretation was maybe it's because our pancreatic progenitor stage is not optimal, right? And let me take you back to the developmental stages, right? So we take human pluripotent stem cells, we differentiate the germ layer that gives rise to the gastrointestinal tract, right? And then we pattern these cells to become pancreatic progenitors. And typically, you know, we have, in some cases, the majority of our pop cultures is expressing NKX611 and PDX1. And we know that when we transplant these cells into immunocompromised mouse models, the cells give rise to beautiful islet-like clusters. They give rise to all the hormones that you will find in the other pancreas, glucagon, insulin, somatostatin, pancreatic polypeptide, as well as cytokeratin 19 and trypsin. So they give rise to both exocrine and endocrine pancreas. But if you take a closer look and you perform single cell analysis of this graft, you also see that even the pancreatic progenitors transplanted into mice gives rise to these enterochromaffin cells. So it's not just what we are giving the cells during endocrine commitment in vivo. Even before that step, the pancreatic progenitors itself, it's actually already skewed to give rise to these enterochromaffin cells. So another question that we had was like, okay, what if we purify these pancreatic progenitors and then sort the purified population? And so back in 2017, we identified a marker that can mark a surface marker that tracks with the pancreatic progenitors. It's called GP2. We know here it's images from developing human pancreas performed by Marcella Brissov and Al Power at Vanderbilt, showing that GP2 positive cells co-express PTF1A and NKX611. And we demonstrated that in vitro, GP2 arises just before NKX611. You see that day 10. And then as the cells start to express NKX611, here we're monitoring NKX611 with with the GFP reporter. We see that NKX611 and GP2 are co-expressed. And so what we wanted to know was like, if we sort for the GP2 positive cells and transplant GP2 positive sorted cells, can we actually get rid of the enterochromaffin cells? And the short answer is no. Even in the GP2 positive cells, we still give rise to enterochromaffin cells. So the take-home message here is that what we think is that we are generating pancreatic progenitors. They express PDX1 and NKX611, but the cells retain a certain level of plasticity. And so they're still able to go both to the pancreatic endocrine progenitor population that gives rise to the pancreatic endocrine cells that you normally find in the other pancreas. But alongside this, they retain this plasticity to give rise to enteroendocrine progenitors. And because of the culture conditions that we have, these enteroendocrine progenitors give rise to enterochromaffin cells in vitro. So if we control steps to generate progenitors and the specification of these progenitors, we should be able to direct a better population for future use in the clinic. And this is work that a wonderful clinician fellow in the lab, Dr. Paresh Mishra, has been really try to pursue, and it's to limit or reduce the frequency of the SLC 18 positive cells that are marking these enterochromaffin cells. You see at the top was in our old protocols was generating approximately 20% total SLC positive cells. we're now being able to drop down this number, which is an indication that this can be effective. So we're working towards that. Just to confirm that the C-peptide positive cells is monomolar, you see approximately 44% of the C-peptide is non-expressing glucagon. There is roughly 20% of the cells that are polyhormonal, and we know this will give rise to alpha cells in vivo and in vitro.
Quick question. Someone is asking from Boston. I hope I haven't overlooked. How do these cells functionally respond compared to cells that develop in the presence or absence of macrophages?
Yes, so the macrophages does not change the frequency of beta cells, and the functionality is the same. Yeah, so it's not improving function at this point. We might have to take them a bit longer in vitro because especially the group of Timo Tonkowski has shown that if you grow the cells for several weeks, you can improve glucose-stimulated insulin secretion. We've only analyzed them at the early stages in the presence of macrophages. We don't see changes. I don't know if it's going to change in after extended culture, but we have to try. Yes.
And then there's one question: the amount of macrophages appear in co-culture more than the ones in the staining. Could you get a clearer phenotype if you reduce the macrophages in the co-culture?
Yeah, it's something that we always think about. Clearly, we have an unphysiological number of macrophages in culture, and this is something that, it seems to be a requirement in a way. We need to have a lot of macrophages in our culture condition to see the effect that you see. So it's not what happens in vivo. You have very few macrophages. Yeah, that's that. Yeah. But if they're useful and they could promote endocrinogenesis and support a much healthier culture, it's definitely something that is worth exploring, especially, what is the mechanism. I think Adriana has done some experiment at the very beginning of testing different number of macrophages, and she identified, she's at the beginning, she's mixing 20%, if I'm not. You can check because this is in bioarchive at the moment, so the number of macrophages that we use is there, but it's whether at the later stages we could use less. It's a question that still remains to be addressed. Yeah. And I'm almost at the end because this is just, to summarize, is a way of saying, yes, we can control the endocrine commitment. And I think there is a way of control the specification of different hormone types. And ultimately, I think this will be important moving forward if we're going to use genome-edited cell lines to generate a transplantable product in the absence of immune suppression.
Yeah, it's definitely a great, new approach, and there's, of course, with any sort of new approach, there's room for finessing protocols.
Exactly.
And then from New York, do you have spatial microscopy data showing close interactions between the macrophages and T cells?
We haven't looked. Yeah. But we can easily look at that. Yes.
Sounds like a collaborative conversation.
Yeah. I need to open the chat so I can Remy. Something to look at. So in summary, I think what I've shown you is that the identification of these macrophages that seems to be possibly developmentally controlled, or at least a phenotype of the macrophages. And what we've shown is that if human ES-derived macrophages that resemble the primitive yolk sac-derived macrophages can impact endocrinogenesis in vitro. We also have data, and you can look at the paper, that it seems that the co-transplantation of these macrophages with endocrine cells promotes survival and engraftment of the cells. And that could be related to the fact that macrophages are known to promote angiogenesis and recruit blood vessels. And this is also an active area of study. And last but not least, I show you just a little teaser on the fact that while we think that we're working with the stage 4 pancreatic progenitor cells that is still plastic, maybe we haven't had enough time to lock the cells in the pancreatic progenitor's lineage. So somehow when they're specified to the endocrine differentiation, or even in vitro, these cells can still go into formation of enterochromaffin cells. But we can, by modifying early and later stages of development, limit the number of enterochromaffin cells generated. And last but not least, the most important slide is actually my thank you slide to all the people who did the work. I mentioned Adriana and Paraish, but also huge support from Farida Sarangi, Angel, and Alex, who's a wonderful PhD student in the lab, who's generated some of the beautiful graphic that you saw in this PowerPoint presentation. A big thanks to the organ donors and their families and our collaborators and funding agency. I'm happy to take questions if you have more. Thank you.
Yeah, we had plenty of questions on our, during the context. So thanks for sort of like being peppered with questions as you tried to give your talk. But yeah, this is very beautiful work and really an interesting new, idea and way forward, thinking about how macrophages can, promote and keep things healthy, in the implant space. So, really looking forward to what's coming next from your laboratory, and we will certainly be watching closely and perhaps reaching out to Dr. Migliorini. Sorry. Yeah, to discuss more about her findings. Thank you so much, and thank you all at McEwen and University of Toronto for sharing this beautiful work with us today on November 14th, Global Diabetes Day. Thanks for everything you do.
Thank you so much, Monica, and everyone.
Have a great rest of your day. So many accolades in the audience for you. So many claps.
Thank you so much.
Bye-bye.
Bye, everybody. Bye-bye.